Hey Isabel, do you remember the viral ALS Ice Bucket Challenge back in 2014?
Was that the trend where people were pouring buckets of ice over their head? Yeah, but actually, what was the point of the whole trend? Like, what does pouring buckets of ice over your head have anything to do with ALS? To be honest, I only know because I saw Taylor Swift do the challenge.
Well, ALS stands for amyotrophic lateral sclerosis. The challenge sort of symbolizes the physical discomfort and muscle stiffness experienced by ALS patients. It's sort of like that weird feeling you get shortly after cold water gushes over your body.
Damn, but is it really a serious condition though?
From what I've heard, the disease can occur at random with no family history or clearly associated risk factors. And it's a progressive motor neuron disorder that is ultimately fatal. Its initial symptoms are limb weakness, twitching, difficulty swallowing, and even speech impairment.
Oh wow, that sounds pretty bad. Is there a cure for it then?
Not to be a nerd, but I was actually just reading a paper about this. You see, there's currently no effective cure for ALS, just a few approved types of medication that prolong survival. I got interested in microbiota after cutting up our SCOBY for that one microbiology module, and found a paper that explored the associations between gut microbiota and various neurological diseases, possibly ALS.
Oh, that sounds interesting. I mean, we did learn that the gut microbiota is engaged in metabolic regulation and immune defense, right? Through the interplay with multiple organs and systems beyond the intestine?
Yep. Recent papers actually highlighted that our gut microbiome contributes to neurological diseases like Alzheimer's and multiple sclerosis. Apparently, things can leak out of our intestines and cause diseases in other parts of the body. For example, some metabolites produced by gut microbes can inhibit responses in our central nervous system. One study even found that a change in our gut microbiome can lead to motor neuron dysfunction, which shows how an imbalanced gut microbiota can potentially cause ALS.
Whoa, I'm mind blown.
Interesting, right? Despite all this evidence, there was no conclusion about the causative role of the microbiome in ALS. This paper works like a stepping stone towards that by comparing the gut microflora between ALS patients and their healthy family caregivers. Using metagenomic sequencing and metabolomics, they established associations between the two.
Wow, can you tell me more about this study?
Sure. Let's start with the study aims.
The researchers' main aim was to determine the discrepancies in gut microflora diversity and associated metabolism between ALS patients and their healthy family caregivers to ultimately provide further evidence to assess the causative role of microbiome in ALS. The team enrolled 20 eligible patients who were clinically diagnosed with probable to definite ALS. They also recruited 20 healthy match controls living closely with the patient to minimize the effects caused by different lifestyles and diets. Also note that this study was conducted in Hunan Province, China, and that the sample size was rather small, making it unrepresentative of not just other regions in China, but possibly the rest of the world. It is important to note that there were quite a number of exclusion factors for this study, for example, multifocal motor neuropathy, which is a muscle disorder and various GI tract diseases. Exclusion factors may be confounders for the outcome parameter, which would be the causative role of microbiome in ALS, so excluding these would produce stronger results. For sample collection, fresh 2 gram stool specimens were obtained from both the patient group and the healthy control group. These sample tubes were frozen in liquid nitrogen and transferred to a negative 80 degrees Celsius fridge for preservation until the next sequencing steps. This holds all microbial activity and ensures that the subsequent uncovered microbial profile reflects the true state of the gut microbiome.
16S RNA sequencing, metagenomics, and metabolomics analysis were done on stool samples from individuals with ALS and healthy controls to gain insights into their gut microbiota. In 16S RNA sequencing, researchers focused on the V4 region of the prokaryotic small subunit 16S RNA gene. The region was amplified, quantified, and sequenced. The resulting data was then categorized into operational taxonomic units. This method helps researchers understand the connection between the composition of microorganisms in the stool and ALS. However, this could only provide information about the microbes up to the genus level. Thus, they conducted shotgun sequencing on the samples as well, which sequences the entire genome of fecal microbes up to the species level, allowing for more detailed examination of the specific types of bacteria and their potential roles. Researchers also combine metagenomics with metabolomics. Metabolomics identifies and analyzes metabolic products. By studying changes in the metabolite composition, they gain deeper insights into the development of ALS. This approach emphasizes the importance of considering metabolic effects alongside the types of gut microbes when searching for potential ALS treatments. This holistic approach provides valuable insights for exploring novel therapeutic strategies for ALS.
Here are the main results from the study. Firstly, 16S rDNA sequencing and LEF-SE analysis showed a change in gut microbiota composition in ALS patients. Secondly, CAG analysis showed that there was potential dysregulation in gene activity and gene pathways. Lastly, metabolomics and metagenomics analysis showed a correlation between gut microbiota composition and metabolite regulation in ALS patients. Let's look at the 16S rDNA sequencing results.
From the OTUs obtained from 16S rRNA sequencing, a Cha-1 verification curve was constructed. The Child-1 index primarily shows species richness, and the results based on Figure 1a show that ALS patients have greater species diversity in their gut microbiomes as compared to the control. The plateaued ends of the curve shows that the data obtained from the sequencing is reliable and most of the microbes are accounted for within the OTUs. A Shannon index plot was also constructed to compare alpha diversity of the samples between ALS and control patients. The differing height and width of the box plots of the ALS and control samples show that generally both phenotypes have significantly different alpha diversity. To measure beta diversity between samples, two PCOA plots were constructed based on unweighted and weighted unifrac. As seen in 1C and 1D, whereby all the samples are relatively close to each other and within the overlaps, it can be said that any differences due to phylogenetic relatedness are not significant. Collecting the data over on a Venn diagram in 1E shows that the OTUs between ALS and control are fairly similar, but there are a significant number of exclusivities, with ALS having 68 more exclusive species than control test subjects.
Furthermore, by mapping 16S rDNA sequences with RDP and NT16S database, there was an observed increase in bacteroidetes and decrease in firmicutes at the phylum level for ALS patients. A lower FB ratio suggests microbiota dysbiosis, which reflects the undermined health in patients with ALS. Taken as a whole, this indicates that the differences between an ALS and a normal microbiome was significant enough to warrant further analysis. To identify the taxonomic cliques that are differential in abundance between ALS and control, LEF analysis was conducted and LDA scores were obtained. The LDA score indicates the extent to which the corresponding group is affected by the differential microbes. As shown in green, ALS patients have a high difference in abundance in these four features. What the team suggests was that these features with high differential abundance could be deemed as potential biomarkers to predict the risk of ALS. Taken together, the results suggest that the composition of gut microbiota was indeed affected in ALS patients. Let's look at the second finding on potential dysregulation of gene activity and gene pathways.
The metagenomic data from shotgun sequencing was cleaned and aligned to form unigenes, which are mapped out onto a volcano plot as seen in 3A. From 3A and B, it can be seen that most genes did not have significant change in activity, and the majority of genes with significant change in activity were upregulated in ALS patients. Cross-referencing this data with 42 gene pathway categories found in tick pathway libraries, revealed that 29 gene pathways had a change in activity as seen in 3E. The bigger the dot, the greater the number of differentially expressed unigenes in that pathway. The risk factor refers to the ratio between the number of differentially expressed genes to the total number of genes in that pathway. And the higher the risk factor, the greater the degree of enrichment. Note that some of the pathways impacted were disease pathways such as RA and malaria, which shows that ALS patients may have dysregulated gene pathways that could lead to greater disease predisposition.
Lastly, a comprehensive analysis combining both metabolomics and metagenomics showed that there was a correlation between altered gut microbiota and metabolite regulation in ALS patients. The PLS-GA score chart, which is a PCOA plot with linear regression, showed that there were intra-group similarities and inter-group differences in metabolites between ALS patients and control. This figure shows the network of differentially expressed species and its correlated metabolites. Green triangle represents species and the purple dot represents metabolite. On the negative ion mode on the left, there were 2 positive correlations between species and metabolites. As for the positive ion mode on the right, there were 2 positive correlations and 1 negative correlations. From the Keck's autology entry, it is known that Enterococcus columbae contains genes involved in fructose and mannose metabolism, amino sugar, and nucleotide sugar metabolism. The Halodaptus species, on the other hand, contains genes involved in the regulation of fatty acids by contributing to galactose, glycerolipid, and sphingolipid metabolism. However, since the study was unable to identify if the alteration of microbes and metabolites was before or after ALS, the relationship was only a correlation.
So, is this a perfect study which solves many questions about ALS?
Well, aside from poor sampling, this study is also a correlative study, meaning that we can't say anything about the causes of ALS.
What do you mean?
Well, correlation does not mean causation. It's like the chicken and egg question. All we know is that some bacteria are enriched while others are suppressed in an ALS patient. But there's no way to tell if the changes in the gut microbiota came first and caused ALS in this person, or if the ALS came first and caused the changes in the gut microbiota.
I see. Then, isn't this study a bit useless?
No. This study helps us to narrow down targets for future studies by revealing the relationship between certain bacterial types and ALS. All that's left for scientists to do now is to answer the chicken and egg question. And this can be done by introducing certain bacterial types into animal models and seeing if ALS is induced in them, or they can induce ALS and see if their gut microbiota changes. Of course, this could be complicated and might take a while, but if we can reveal the mechanisms behind ALS development, it will give us many targets for drug development, hopefully preventing and even curing ALS. My gut tells me that the future of ALS research will be very interesting. Thanks for listening to our presentation.
We nominate Junhong, Chuling, and Chunwen for the ALS Ice Bucket Challenge.